It's almost
Sorry about the delay.
They already
Studying for it, though. When you take it, you can't use your notes.
Everybody, good morning.
this cool.
We can study and do all the questions.
Oh, cool.
So I'll be talking today about gene therapy and genome editing. Please feel free to jump in with any discussion, questions, or anything like that during these slides. The main problem that I'll be discussing today is on the issue of human genetic disease. Gene therapy is also used for a number of other applications, cancer immunotherapy. I'm not going to focus on those today, but I'll be speaking specifically about how these methods are used to try to address inherited genetic disease. There's over 75,000 different genetic variants have been already characterized to cause disease in humans. I'm showing here a pie chart of how the genomic aberrations are sort of divided between these. About half of pathogenic variants in the genome are caused by single nucleotide changes. 30% of them are transition changes, which are based on purine to purine or pyrimidine to pyrimidine changes, 20% on transversions, and then 26% on small deletions, 8.2 on duplications, and then less on other larger aberrations like translocations or copy number gain and loss. In total, over 1 in 50 newborns are born with some kind of heritable genetic disorder that impacts their life. And at the moment, there aren't any FDA-approved therapies that directly reverse the root cause of disease, but these are now excitingly in clinical trials at the moment, and so we're expecting things like that to come very soon. But based on the gene that's mutated and the type of variant that you have, of course, you can have super different manifestations of disease, ranging from accelerated aging for progeria, sickling of red blood cells and sickle cell disease, mucus buildup in the lung, and there's CNS degeneration from Tay-Sachs disease. And for every disease, every organ that's affected, you typically have to have a different strategy of treating that kind of disease. So we'll go over some of those strategies in the discussion today. And here's an overview of what I'll talk about. So a little bit of the origins of genome editing, what gene therapy, traditional gene therapy by gene addition is like, and current gene therapies, how we use nucleases to edit. I'll talk a little bit more about the one FDA-approved genome editing therapy, sickle cell disease. I'll talk a little bit about base editing and prime editing and how we've been using those. This is sort of my area of expertise and research, so I'll be discussing some of the papers we published in this space to give you an idea of what research in that area looks like. Talk a little bit about methods for in vivo delivery in that now. So, how did we first come to know that DNA is what we need to change in order to treat genetic disease? And that had been a question a long time coming. It was really in 1944 when we had this first inclination that nucleic acid chemical moieties are the transforming principle that allow phenotypic changes in bacteria that are injected into mice. So looking at these different rough and smooth strains, some of which kill mice, the researchers here were injecting nucleic acids versus protein components and saw that it was the DNA that was important to confer the phenotype of these bacteria. There's another famous Hershey-Chase experiment looking at how viruses transmit their genetic information, and that came through DNA, nucleic acids, rather than protein. And then in 1953, Watson, Crick, Franklin had uncovered chemical structure of deoxyribonucleic acid. They postulated this as the structure, which has since borne out to be true. They mentioned how it does not escape their notice that the specific pairing that they postulate immediately suggests a possible copying mechanism for genetic material, which of course turned out to be correct in how DNA is polymerized. So now that we know what DNA is and its structure, it's already a revolutionary insight, but there's a lot more questions after that. How exactly is more of it made? How does one genome instruct the formation of so many different kinds of cells? How does DNA sequences, how do different DNA sequences lead to various phenotypes? And how is your DNA maintained if it's broken? There's so many more questions we need to explore. And so there's a little bit more tool development that had to happen before we could start answering some of those. Berg, Dan, and Nathans, late 60s and early 1970s, had identified restriction enzymes from bacteria that can cleave double-stranded DNA and allowing us to map genetic positions even without explicit mutations, so finding out where the right recognition site for a restriction enzyme falls allows you to do this type. And then Gary Mullis in 1984 had developed the first PCR reactions, which I think were a lot more laborious than we do today with the thermocyclers, since they have to transfer these tubes between water baths. But now, with these two tools in hand, you could actually conduct cloning and put your own DNA sequences that you're interested in maybe not your own, maybe bacterial or other DNA sequences that you're interested to study inside plasmids by PCR amplifying one piece using restriction enzymes to cut and paste them together. Now we can actually study how different forms of DNA impact phenotypes of cells. Next thing I wanted to briefly touch on is how we learned about modifying endogenous genomes. In 1974, Yinch and Mintz found that using SV40, this is a viral virus, that they could inject this viral DNA into a mouse blastocyst. And then once you let that mouse grow up into an adult, they sequenced the different tissues of that mouse and found multiple copy numbers of that viral genome in the mouse, showing that DNA could integrate into mouse cells. It would replicate with cells and be expanded. So there was some way that they were modifying the mouse genome to include this viral DNA. Hinton and Fink saw in 1978 that they could take a yeast strain that was missing the LU2 gene, so now they were dependent on leucine to grow. Typically, yeast can make their own leucine, but that relies on this gene LU2. So if you knock that out in cells, you can actually repair that gene back to a LU2 positive state, having a healthy leucine 2 gene using homologous recombination. So you encode that healthy gene in the plasmid, and it has a lot of overlap with the natural LU2, except the plasmid does not have a mutation. It just has a single base change that disrupts the gene. And you can integrate that whole plasmid into the yeast genome and fix the condition. And now this is a convenient selectable marker because cells that undergo this process will also be able to make their own leucine and able to grow on media lacking leucine. So people use this trick to deliver any other gene that they want by hooking that up to a healthy leucine gene and integrating it alongside leucine. You select the leucine positive cells, and those also have the kind of DNA that you want to study integrated inside. And then in 1981, Orweaver, Shostak, Rothstein discussed how different homology arms, so amounts of homology to the genome, can really change DNA repair efficiency. And saw that if you made a DNA gap, double-stranded break, made synthetically in a plasmid, that you can vastly alter the DNA repair outcome distributions. I actually had the fortune of overlapping at the Broad Institute, or at the Whitehead Institute during my PhD with Danish, Fink, and Orweaver. They were all there as well, so doing research at the time. It was nice to get to know some of these pioneers in genetic technology. Thomas Capecci and others then saw, so unlike in yeast where homologous recombination is really efficient, you can inject any DNA and expect some of it to get integrated. In mammalian cells, like mice, homologous recombination is not the dominant repair outcome after a double-stranded break. So other methods are needed to really get precise repair in mammals. One other useful tool I think that was developed in the 80s here I want to discuss is pre-recombinase. So pre-recombinase stands for causes recombination. It's a protein that was identified in the P1 bacteriophage, and it caused very efficient integration of DNA sequences into the genome. And it relied on these recombinase sites, relied on these LOXP sites in order to integrate genetic sequences between that recognition sequence. Depending on the orientation and distance of the LOXP sites, you can get a variety of outcomes. So if you have two that are arranged opposite to one another, you can invert the sequence between them. If you have two that are arranged in tandem, then you delete the sequence between them. And if you have two that are on totally different pieces of DNA, you can swap the regions that are distal to the LOXP site. So this allowed a whole new set of nucleic acid acrobatics to be done in the genome to try to probe genetic phenomena. Then in 1984, there was another study, Williams, Lashemka, Nathan Milligan. They showed that you can use an antibiotic resistance cassette integrated into mouse hematopoietic stem cells. So they would deliver this with a virus into hematopoietic stem cells of a mouse and graft these back into the mouse. And they would show that even after a secondary transplant, where you took the bone marrow from that first mouse and put it into a second mouse, you can maintain modified blood. These are the kind of plots that we're getting into nature at the time. Not super clear, but you can still see bands from this recombination, from the antibiotic resistance, even in the secondary bone marrow transplantation. Just proving that they can maintain the stem cell lineage that has been permanently modified in a desired way. Now, antibiotic resistance can be useful in some circumstances, but we really want to be able to modify blood to treat blood disease. So that's where a lot of research is focused since then. So first I want to overview how modern gene therapy has been working based on those historical insights. One major source of gene therapy has been lentiviruses, or retroviruses. These can integrate healthy genes into patient cells, and this is typically done in the blood because those cells can now be retransplanted and grafted back in the patient, just like what Williams had done in mice. So this is used to create anti-cancer CAR T cells. Now it's been approved to add healthy hemoglobin to blood. So if you have the sickle cell variant, or if you have a deletion in your adult beta globin gene, you have sickle cell disease or beta thalassemia, and either of those conditions can be fixed by the addition of a healthy beta globin gene back into blood cells. The advantage of this kind of method is permanent addition of the desired gene. So once it's integrated into the genome, all the progeny cells will contain that gene and will be there throughout the lineage of those treated cells. And you can get very efficient transduction. More than 90% of cells in your dish can be modified to contain the viral gene. Showing here the process of what happens. So you harvest cells from the patient, culture the stem-like cells, the CD34, those that have the CD34 marker on the surface, which are hematopoietic stem and progenitor cells, or HSPCs. Then in vitro, you culture those along with your lentiviral vector that contains beta globin. That's integrated into the genome, and now you reinfuse that patient after ablating their endogenous bone marrow. So one of the most challenging parts of this therapy is that you have to treat this patient with radiation or chemotherapy to destroy their endogenous bone marrow, which is a painful and difficult process. But after a few weeks, then the engrafted cells will reconstitute their blood system, and they should, at that point for life, have a healthy hematopoietic system. There's some disadvantages of this approach. So there's always a risk of oncogenic insertional mutagenesis. We can't perfectly control where that lentiviral genome is integrated into the human one. And if it goes into a proto-oncogene or a tumor suppressor gene, then it can disrupt its expression and increase the likelihood that the patient will get leukemia or some other kind of cancer. The transgene can be silenced over time depending on the location that it falls and the regulatory elements that you choose. I was actually surprised just last month I got a call from some physicians at MGH Hospital who were treating patients that were treated in this manner, and they were reporting that maybe 5 years after treatment they started to get more pain crises, vaso-occlusive crises, so they wanted to discuss the causes of that, how the engraftment goes, and whether the new therapies could help prevent that. It's generally not used in vivo with a direct injection of the lentiviral vector into the patient, because that can stimulate a very strong immune response. This is maybe exemplified best in this unfortunate situation, and a tragic situation really. In 1999, an 18-year-old Jesse was treated with a recombinant adenoviral vector, a candidate for treating a genetic condition by delivering healthy ornithine transcarbamylase to the liver. And though his disease was very mild, he had volunteered for this treatment so that he could help bring it to patients that are more severely affected and really need it. However, he wasn't informed of the risks of the vector, or that 3 of the treated monkeys had died from liver inflammation following that treatment, even before his administration. He also experienced this cytokine storm, severe immune response, and died 4 days after receiving the treatment. So this incident is well known in the field as something that really raised a lot of caution about gene therapy. People say it's slowed down gene therapy for a decade, but I think there's a lot of good reasons to actually slow down when you're developing something new like this, and try to be very careful. You want to avoid tragedies like this when you're delivering any kind of reviral vector. However, so that was with adenoviral vector. That's a double-stranded DNA virus that can stick around for quite a while in the liver. People then realized that adeno-associated virus, which is a much smaller virus, is much less immunogenic. So you can inject this virus in monkeys and people, you don't get substantial immune responses, partly because this virus is not known to naturally cause disease. So it seems like it's largely silent in the body. So this AAV vector has a single-stranded DNA genome, and it's used to episomally maintain healthy genes that you can deliver by recoding its DNA. So the advantages of AAV are that you can actually inject them in vivo. They have low immunogenicity. They also have low mutagenicity because integration into the genome is extremely rare with these vectors. But they do come with a number of disadvantages as well. So there's cargo size limit of 4.7 kb. So with the adenovirus, you can add more than 30 kb of genetic sequence, which is for multiple cassettes and genes. 4.7 is quite limiting. There are genes we want to deliver that we just can't fit into an AAV. The duration of expression may be limited. This is actually just the same as an adenovirus. Both adenovirus and AAV, they're maintained episomally, so it doesn't integrate into the cellular genome. Cells don't replicate the vector when they're dividing. This is very different from lentiviral integration, where now this lentiviral cassette is in the genome, gets replicated with cells. If you do lose effectiveness of your AAV therapy over time, you likely cannot re-administer these. All current trials have as a condition no former treatment with AAV, because even though they're generally immune silent, when you inject a massive concentration like you need for gene therapy, then your body does still raise an immune response against the capsid. So it's thought to be extremely dangerous to try to re-administer an AAV therapy. In fact, I don't think it's been done yet. Maybe there are ways people are talking about adding drugs that will temporarily knock out the immune system and proteases that will cleave all the antibodies in a person's blood to try to avoid immunogenicity, but at the moment it hasn't been done yet. And one last thing is it's very difficult to simulate endogenous gene regulation. So if you have a disease and a gene that's very carefully maintained in your cells, when you add that healthy gene back in with AAV, typically it's massively overexpressed. It doesn't have the same enhancers, repressive elements that might be located next to it in its broader context in the genome. Now you're expressing a whole lot of this healthy gene immediately. And over time, if cells divide and this vector is the episome is lost, then you might decrease that expression. And so you get a change in expression over time that's dependent on the vector and not dependent on what the body really needs, which can cause a problem. So I'm showing here a strategy used for the very first gene therapy used to treat a genetic disorder, which is later congenital amaurosis, which is a form of blindness caused by mutations in the RPE65 gene. The protein that this gene forms is involved in the retinal cycling. It's a necessary part of the visual cycle. So if you don't have this, then every photon can essentially fire. Every photoreceptor can fire about once, and then you can't do it again because you don't have any more of the small molecule you need to absorb light. And so this can be treated by delivering a healthy RPE65 gene to the cells that use this. So this is an AAV2 vector to the retinal pigmented epithelium. So they inject this subretinally. The AAV transduces the pigmented epithelium at the back end of your eye. Now these cells can express the healthy gene and cycle that retina out. These patients can see. In 2019, Zolgensma was approved. This treats spinal muscular atrophy that's caused by SNM1 variants. And this is affected by using a different serotype of AAV. So many different AAV capsid sequences have been identified naturally in the population. AAV2 and AAV9 are two different capsid serotypes that each have different transduction efficiencies. So AAV2 is particularly good in the retina. AAV9 is particularly good at neurons. And this can target neurons in the spinal cord when injected intravenously. 2022, 2023 saw the approvals of Hemgenix and Roctavian, which treat hemophilia B and A respectively. These deliver healthy factor 9 and factor 8 genes, which are involved in the blood clotting factors. This used AAV5, which is particularly efficient at transducing hepatocytes. Hepatocytes naturally express these genes.
So has there been enough follow-up research to see like decreased efficacy? You know, I mean, now we have 8, 6, and 3 years later.
Yeah, so I'm not aware of that with Zolgensma yet, though I know this has never been fully efficacious to totally, eliminate disease. It reduces disease significantly with Zolgensma. With Luxturna and both of these, I have seen reports that there's decreased efficiency in 5 years later. Patients start expressing less of these genes, and their vision decreases, their hemophilia relapses, so there are issues, and then you can't treat the patient again. These treatments all cost at least $500,000, typically more than $1 million, so it's a large investment, definitely valuable to get 5 years of healthy life, but it's a recurring problem. That's not a permanent treatment for these conditions. And then in 2023, Elevidys treats Duchenne muscular dystrophy. It delivers an engineered dystrophin gene in this RH74. This was a serotype actually identified in monkeys, thought to be less immunogenic in humans. Do an intravenous injection into patients to deliver a miniaturized dystrophin gene. There's also one in vivo gene therapy that has been approved for topical use, so just applying it with a gel on the skin. This is for treating blistering skin disorder, recessive dystrophic epidermolysis bullosa. This, unlike AAV or lentivirus, this uses a herpes simplex virus, which is a double-stranded DNA virus, also maintains its DNA episomally in transduced cells. This resulted in this approval by Quebec in 2023. This delivers a healthy collagen gene, which is the gene that's mutated in patients with this epidermolysis bullosa type. And if you apply this gel to blistered skin, then the virus is thought to be able to get into the dermal epidermal junction cells and start expressing this collagen, which is what you need to actually anchor your dermis to epidermis. Without that, then you get massive blistering all over your body. Unfortunately, these patients suffer from a lot more than just external blisters. The skin or they have dermal epidermal junctions in their mouth and other parts of their body as well that are also painful due to this disease, and there's no treatment for that at the moment. So, I talk now about pretty much all of the gene therapies that are approved for use now in humans based on the addition of a healthy gene. So traditionally, gene therapy means just that addition of a healthy gene. More recently, that definition has expanded to any therapy that will correct or introduce a gene, so including genome editing as a therapeutic strategy. I want to talk about how this genome editing strategy is a little bit different than gene addition, where you just deliver the healthy gene. So using genome editing, you can restore a healthy cellular genotype and even restore native regulation of that gene. The therapy, the medicine itself, only has to be delivered transiently because you don't really need to permanently express the genome editor once it's done its job and fixed the genome. You can express it for a little bit, and then the edit's permanently there, and the native gene getting replicated with cells, just like lentiviral integration. If your stem cells are corrected, then all of its progeny will also be corrected, and so the expression should not be variable or diminish over time. Unless you're not editing the stem cells, in which case the unedited stem cells will slowly replace the edited ones, and you would see that drop in efficacy. There's a lot of disadvantages we need to consider as well. So genome editors aren't natural human genes, and they might elicit their own immune response that a healthy gene would not. There's always a potential when you're targeting one site in a huge multi-billion nucleotide genome that you could actually get off-target edits, increasing mutations at other sites in the genome. So both for this propensity for off-target editing and propensity for an immune response, it's really undesirable to get durable expression. You'd rather express the genome editor very transiently, have the effect you want, and then have that edited go away. One other complication, you might need to make a new medicine every single time you have a new variant that you want to correct. So unlike gene therapy, where you can deliver a healthy gene, and any variant in that gene can be treated with the same medicine, if you're using editing to directly correct a mutant variant, then you need a new medicine for every single new form of therapy for every single variant. Some diseases have over 1,000 variants known to cause genetic disease, so that would be quite a lot of medicines to produce. So at the moment, development of genome editing therapies that is currently being focused on by companies or in clinical trials are either for cases where you're not actually correcting a specific mutation, you're disrupting a different gene, so because of that, doing some genetic trick so that you're able to treat many different genotypes. And there's 2 trials ongoing where you are actually directly correcting a genetic variant, only in cases where there's one variant that's very common in the population. So if there's a founder variant and this is causing more than 30% of disease, then companies have found that economically practical to actually try to correct it. So a little bit about how genome editing works. I showed earlier on that it was identified you could vastly stimulate homology-directed repair if you make a double-stranded DNA break, but that's not the most common outcome in a million cells. So when you make a double-stranded break, it's possible to add a healthy sequence, a template for homologous recombination, where you perfectly restore the genetic sequence to any desired outcome, fixing a genetic mutation. But the most common outcome in human cells is NHEJ, non-homologous end joining, involving these DNA repair factors. And this is a much more messy process where you'll get stochastic insertions or deletions. If you're targeting a diseased gene, you want to fix it. If all or 90% of your resulting alleles have new mutations, then you're not really helping very much. So because some of these factors involved in homology recombination, especially RAD51 and BRCA2, these are cell cycle dependent genes, this is particularly inefficient in vivo. So when you're working with rapidly dividing stem cells in a dish, mouse embryonic stem cells or iPS cells, you can get more than 50% homology-directed repair because these cells are dividing quickly. But thankfully, cells in you and I aren't dividing every day, or else we'd be doubling in size every day. So cell division in vivo is actually quite slow. People haven't really seen in vivo HDR of more than 5%. So not enough to correct most genetic diseases. And that's why most of the in vivo editing using a nuclease is focused on this pathway. I'll show one example of that. I'll show one example of that in the following slide. First, I want to go over how these double-stranded DNA breaks are introduced. So for several decades, we've known about meganucleases. I believe these were first found as a homing endonuclease from yeast that's involved in mating-type switching. These can very efficiently cleave target sites in the genome. However, they have 18 or more nucleotide domains that they recognize and bind. It's extremely difficult to reprogram these nucleases to cut other sequences. You're essentially limited to naturally occurring sequences that you want to cut. And in the human genome, I believe there's no cut sites for athg, the very first homing meganuclease that was identified. So it was useful for some strains. If you somehow got a human cell with an ISC1 site in it, then you could target that for editing. But you can't have that versatility to target any gene you want. This issue was partly solved with zinc finger nucleases. These are a lot more modular. Zinc fingers are, I believe, the most common DNA binding motif naturally in humans and most organisms. So they underlie a lot of transcription factors. Naturally, they're not nucleases, but some clever researchers figured that they can fuse the split FOC1 domain. So half of it on its own isn't competent to cut DNA. Only when two halves are brought in close proximity can they reconstitute the full enzyme and make a double-stranded break. And so if you target two different zinc finger domains nearby one another, when they bind to off-target sites, you won't get an edit. When they bind to that target site, both at the same time, they'll make it a double-stranded break. So this is much better. Some of these fingers, most zinc fingers are formed of 3 individual finger modules, and each of those modules recognizes a 3 base pair sequence. There's some ability to mix and match these as you want to recode them, but they're never perfect. You often still have to do a little bit of directed evolution to get highly efficient cleavage. The modularity problem we all thought was solved around 2009, 2010, when the TALEN code was identified. These are transcription activator-like effector nucleases. They use the same trick of having 2 different DNA binding motifs that you program separately, fuse them to this FOC1 nuclease domain, and then only when both of them bind in proximity at the same time do you get the double-stranded break. And here, there's much greater modularity. You don't have to do any in vitro evolution or engineering. You know exactly which TALEN repeat binds to what nucleotide of DNA. So you have full modularity to target any sequence you want. The problem is these TALENs are each over 100 kilobases of, over 100 kilodaltons, so several kilobases of DNA. You'd have to remake a pretty large gene every time you want to target a new sequence. And so that's why, even though this was a really great tool at the time, it was hard to clone these. In 2012, there was a big breakthrough with the establishment of CRISPR-Cas9 as a gene editing method. And the big advance here is that rather than having to reprogram a whole multi-kilobase long gene, they found that the nuclease can be retargeted just with a 20 base pair change in sequence. So this nuclease now is totally, the nuclease doesn't look at the DNA sequence that it's coding whatsoever. It really just relies on complementarity to a guide RNA that it binds to recognize its target site. This was stimulated the award of a Nobel Prize recently to Jennifer Doudna and Emmanuel Charpentier, who defined this system. I think it's a really interesting story how this was first found, what it does endogenously. It's used by bacterial species to target and cut phage DNA to prevent infection. It's kind of like an acquired immune system in bacteria because bacteria that survive a viral infection can actually integrate 20 nucleotides of sequence from that virus into their genome, and they store that sequence forever. So if their progeny sees that virus again, they have it ready, fused to their guide RNA, ready to target and cut the infected particle. Typically, or endogenously in bacteria, this is actually a combination of two RNAs that hybridize to each other, the CRISPR RNA and the tracer RNA. But in this paper in 2012, they figured out a way to synthetically fuse these together, so you don't even have to separately express two RNAs. It's even easier to just have a single guide RNA or sgRNA to recode, deliver that with your one enzyme protein. And now you have flexibility to cut. They showed in this GFP plasmid they could cut circular plasmid at 5 different sites and get very efficient cleavage. And so, the very first place where we want to apply this revolutionary technology is sickle cell disease. This is the most common deadly genetic disorder around the world, with over 300,000 children born with sickle cell disease every year. It's caused by every individual that has sickle cell disease has at least one copy of this particular variant. It's a glutamate to valine amino acid change at position 6 of adult beta globin. When you have this valine in position 6, under deoxygenating conditions, the hemoglobin can polymerize into very long fibrils. And this causes cells to sickle, change their shape from this nice round shape that gets through blood vessels easily to a curved shape that now clogs blood vessels and can lead to decrease in oxygen to any tissues that are downstream of these clogged vessels. This can lead to death of tissue, to terrible pain throughout the body, increasing risk of stroke, and greatly shortens the lifespan of people that inherit this disease. There are some other beta globin variants that, when co-inherited with this, also cause forms of sickle cell disease. So it's not that everybody has two copies of this allele, but everybody with sickle cell disease has at least one copy of this nucleotide change. I also want to discuss beta thalassemia. So beta thalassemia and sickle cell disease are each among the top 5 most common genetic diseases in the world. Beta thalassemia is caused by loss of function variants in the beta globin gene. So rather than having the single nucleotide change that causes polymerization, now you just don't have your beta globin. Beta globin is meant to form a 1-to-1 molar ratio complex with alpha globin. It turns out when there's not enough beta globin around being expressed, then you have these excess alpha globin chains that would misfold and aggregate into big particles, messing with your red blood cells. Unlike sickle cell disease, where every patient has at least one copy of the same variant, there's a lot of allelic heterogeneity among beta thalassemia patients. So you can have many different null alleles in the promoter and the introns and the coding sequence of adult beta globin. And symptoms can range from mild to severe based on how much loss of function you have. You get systemic toxicity from very poor oxygen transport and red blood cell toxicity, lower red blood cells, and all of the misfolded alpha globin. So the most common treatments for patients that suffer from either of these diseases is to have a regular blood transfusion to get some healthy beta globin red blood cells into their system. Sickle cell disease is most common in sub-Saharan Africa, India, South America. It's where regions where malaria is endemic, because if you're an individual with just one copy of the sickle cell allele, it's called sickle cell trait, and protected against malaria. So there was some positive selection for this allele to be maintained. But the unlucky individuals that inherit 2 of those alleles then suffer from this terrible disease. In the United States, the Black population is most severely impacted with sickle cell disease. 1 in 13 people, Black individuals are harboring sickle cell trait. 1 in 365 Black children born in the United States inherit sickle cell disease. There is a cure for sickle cell disease that's been known for a few decades now, which is an allogeneic bone marrow transplantation. So you take a bone marrow from a family member or from somebody that's matched well enough with the HLA receptors, and then you go through a similar process to what I was describing before. You'd have to use radiation or chemotherapy to destroy the endogenous blood system in the patient. You'd then inject the healthy allogeneic donated cells into the bone here, and over time, the whole blood system of that recipient should be reconstituted by the donor cells. However, this has a couple of major barriers to it. One is that most patients don't actually have a suitable donor available for them. And even in the patients that do, I think it's about 10 to 30% of them still suffer from graft versus host disease, where the new blood system eventually recognizes that the body it's in is not self and starts to target it with the immune system to destroy now the tissues of the patient, which is typically a deadly condition. So, as an alternative to this allogeneic bone marrow transplantation, use of cells from another human, now the focus is more on autologous stem cell modification, where you use cells from the patient themselves, edit them in some way, modify the genome to correct the disease, and retransplant those back in. And here now everybody has a donor, because you can always be your own donor for an autologous stem cell transplant, and you don't have graft versus host disease because you're just using cell cells. And this was exactly the basis of the very first approved, FDA-approved therapy that uses genome editing, which is for sickle cell disease. It's a little bit of a complex mechanism. So it's a nuclease. It doesn't directly correct the genetic variant. They have to use a genetic trick that they found specifically because there's so many people with sickle cell disease. We learned about a lot of modifier genetic loci. If you inherit another particular nucleotide change, you might be protected from sickle cell disease. And so geneticists use this knowledge to figure out that there's a particular enhancer region in BCL11A that's involved in expression of this transcriptional repressor in red blood cells. And so if they target this enhancer region, now you don't get expression of BCL11A in red blood cells. Why is that nice? It's nice because BCL11A binds to the fetal hemoglobin gene and represses it. So if you lose BCL11A in red blood cells, now they express fetal hemoglobin. For any human to be born, they have to have healthy fetal hemoglobin, or they would have suffocated in utero. And so any patient that has inherited sickle cell disease or beta thalassemia can be treated by this method because they'll express their historical fetal hemoglobin that kept them alive as a fetus. So this was just approved not very long ago, December 2023. The trade names are here. So this uses ex vivo electroporation of Cas9 nuclease into hematopoietic stem progenitor cells. And then loss of this repressor gene alleviates fetal hemoglobin repression. Now you get, this is worded wrong, sorry. Alleviates fetal hemoglobin repression, not expression. So now you get expression. So here's a chart of what fetal hemoglobin measurements look like in patients that receive this. So before treatment, indicated by the dotted line here, these patients rely on transfusion of healthy blood. So this HbA in green comes from healthy donors, and it's still maintained for a month or two after the treatment. But eventually, these patients are weaned off of transfusion. You can see most of the hemoglobin in their blood is actually made up of fetal hemoglobin, their own endogenous gene, reactivated again for the first time since they were born. I want to mention why it's important to use this enhancer. So you can envision, there's a thousand, please.
So what is is there just some mechanism where the body says, oh, we have a lot of fetal hemoglobin, it just stops producing the sickle cells? Or does the fetal hemoglobin sort of rescue the phenotype of the red blood cell itself?
So the beta globin gene cluster, fetal hemoglobin, is actually located very close to adult globin in the genome. And there's one enhancer region that is called the locus control region that binds to the genes when they're expressed. And so if you get more expression of fetal hemoglobin endogenously, this wouldn't work if you're expressing it off of an plasmid or something, but if you're getting more fetal hemoglobin expression endogenously, you're actually getting ratiometrically less expression of the sickle globin at the same time. So you get this boost in effectiveness for that reason. And I also wanted to mention knockout of BCL11A really needs to be done specifically in red blood cells. There's 1,000 different ways you could destroy this gene, but if you do that in hematopoietic stem cells, then none of your white blood cells would express it either. And this is an important anti-cancer gene in some contexts, so you'd probably be getting leukemia much more frequently if you disrupted this gene in a general way. And that's why the very key insight here was that there's an erythroid-specific enhancer that only that's necessary for its expression in red blood cells. So this could be disrupted without increasing the risk of white blood cell leukemias. So something I want to talk about now is an alternative method of genome editing that does not rely on making a double-stranded break, but instead directly chemically converts nucleotides to other nucleotides. This was a tool that was developed in 2017, the same year that I joined the lab of David Liu at the Broad Institute by his lab. And so I started applying this tool to sickle cell disease myself during my postdoctoral work. So this uses a deaminase enzyme to convert adenosine to inosine. This naturally occurs in RNA, kind of ADAR enzymes that convert adenosine to inosine. but there's no natural known enzyme that reacts with DNA, converts DNA nucleotides in this way. However, it was a high priority because the opposite nucleotide change, which is a C to T change or a G to A change, this is the most common mutagenic nucleotide change in humans. So almost half of pathogenic point mutations can be corrected by engineering this opposite edit that would convert those A's back into G's, or those T's back into C's. So Nicole Gaudelli in the lab did many rounds of in vitro evolution to take an ADAR, an RNA reactive enzyme, and make it active on genomic DNA. And once she succeeded in this, making this deoxyadenosine deaminase rather than adenosine deaminase, they fused this to Cas9. And the reason for this is we're taking advantage of all of that versatile targeting of the 20 nucleotide guide RNA so that we can retarget our adenine base editor to any region of the genome that we're interested in editing. Now, we don't want to make a double-stranded DNA break, because I already told you that that causes NHEJ, insertions and deletions, and would destroy the gene we're targeting. We make a mutation so this Cas9 doesn't make a double-stranded break, it only makes one-stranded DNA. So here's a little bit more about the mechanism. When you have your guide RNA bound to Cas9, bringing it to its target site in the genome, the guide RNA will anneal to the genome. And when you have the guide annealing, then the strand that originally annealed there is displaced. So now you have a single-stranded piece of genomic DNA that's hanging out from the genome. The key part of these enzymes is they only work on single-stranded nucleic acids. So you're not deaminating nucleotides all over the genome, which is pretty much double-stranded everywhere. You're only doing it right where you're localizing this enzyme to, where there's both a high concentration of enzyme and a high concentration of single-stranded DNA all locally. And so a small window of this displaced R loop, this is called an R loop, with its DNA displaced by RNA. These are substrates for editing. You actually get this chemical reaction, P to I conversion. We keep one of the nuclease domains of Cas9 active, so it nicks the individual strand. And this nick serves as a powerful signal for cells to come and repair that nick. And when it repairs the nick strand, it likes to use this as a template, so that really hard codes in with this A to G change on both strands of the DNA. So now I want to talk a little bit about how these nuclease and base editors are being used currently in clinical trials. I'm listing those that are in clinical trials here using lipid nanoparticles, using Cas9 nuclease to destroy the transthyretin gene. Yeah.
Sorry, on the previous slide, something that you talked about many times before is just when you're making these changes sometimes, this is for base editing is like a transition edit.
Yeah.
So mismatch repair machinery, are there any mismatch repair inhibitors that you can use to I already know it's a pretty efficient process, even without that, but do you think that might improve efficiency as well?
Yeah, I think there's a good chance. It's actually something we're looking at in the lab right now.
Okay.
Some people are doing tests with different inhibitors. I think there are known inhibitors. Nobody's shown a very impressive change at the moment, but there are some new drugs that people are developing all the time, so we're trying to test those and bias repair outcomes even further to get the edit that we want. There's always the chance that the cellular repair machinery can convert the edit back to the wild type state, the original state, which is not what we want.
Yeah.
Great question. There's all kinds of ways you can think of to further engineer the genes or to try to manipulate the cells so that it gives you the editing outcome you want. Some of them might be more dangerous than others. So drugs that manipulate DNA repair might cause higher frequencies of mutations elsewhere. But if you just give the drug temporarily to the patient, then I think that's a great way to, at a very low risk for one day of a little bit lower fidelity of DNA repair, then you can maybe cure that patient for life with a genetic condition. So very worth it. Yeah, thanks.
Can we actually go back to the previous slide real quick? So just to walk through, guide RNA binds. Well, I guess first you have the guide RNA binds. The opposite strand, R loop that's displaced, is then edited.
Yeah.
And then the hope is that the DNA repair will just bias towards the I and replace the T with the C.
That's right. Yeah. So at this stage, you could get editing of either. So this is a common place where the DNA repair machinery will likely convert your I back to an A, and then you'll repair in that way. But because we nick this strand, we see more than a twofold boost in editing efficiencies biasing the other way. It's possible to use a base editor without that nickase. So nicking a strand can increase the rate of indels, so NHEJ repair outcomes, and so especially with C base editors, you can show both of these. So Cytidine base editors have a very similar mechanism, but they use a cytidine deaminase instead of adenosine deaminase. And cytidine deamination produces uracil in the cell, which is also excised because that's a much more common genomic aberration that the cell wants to repair. So if you get uracil excision at the same time as a nick on the other strand, you can get a lot of indels. And so one thing that many labs do is to use a totally nuclease dead Cas9 so you don't nick. That does decrease your editing efficiency quite a bit, but it reduces the frequency of indel outcomes as well. Any other questions about the base editing mechanism? So here I'm listing the in vivo gene therapies that are in trials at the moment. They all involve gene editors. So the first three I'm showing here use a nuclease to disrupt the gene that we want to destroy to treat disease, just like how we're destroying B-cell RNA in red blood cells to increase fetal hemoglobin. Here we're destroying the transthyretin gene, which will prevent more transthyretin being expressed that will lengthen these amyloid fibers and causes disease in transthyretin amyloidosis. And also disrupt this KLKB1 gene to treat hereditary angioedema. These genes are largely expressed in the liver, and so just targeting a lipid nanoparticle containing this editor to the liver, you can vastly reduce the expression of these genes and treat the conditions. They're also using nuclease disruption of the ANGPTL3, which is an inhibitor of lipases, and this lipase itself that will treat high cholesterol. So, I'm sorry, that's not a lipase. I'll have to look at more detail at what that gene actually does. But disruption of both of these genes can reduce cholesterol in individuals. And so, rather than requiring a particular nucleotide change, just destroy a gene that keeps cholesterol around, and you start to treat high cholesterol. And then there's a number of base editing therapies also in trials now already dosed in patients using base editing to destroy the PCSK9 or ANGPTL3 gene, similarly to these can treat high cholesterol. There's two ongoing trials that are actually involving nucleotide correction, where you're fixing a specific variant in the patient. And that's for alpha-1 antitrypsin deficiency caused by mutations in SERPINA1 and for glycogen storage disease caused by mutations in G6PC1. And so each of these diseases have founder mutations that are particularly prevalent in populations that have the disease. So a high enough fraction of patients can be treated by this one nucleotide change that this is now in clinical trials. And then there are also ex vivo base editing approaches that are in trials to treat sickle cell disease and beta thalassemia based on a similar mechanism, so trying to increase fetal hemoglobin and be able to broadly treat this disease. The exact basis of how those drugs are working isn't really published, but a lot of my postdoctoral work in the Broad Institute involved developing these strategies. So I want to sort of walk you through the work that we did there, just so you get a better feel for what we do as we go about developing a gene therapy, gene editing therapy. So for sickle cell disease, an A to G change isn't able to perfectly reverse this pathogenic valine back to glutamic acid. But an A to G change on the bottom strand of DNA can convert that valine to an alanine. Alanine is a rare but naturally occurring variant of beta globin and known to be benign. People are still healthy. They don't have sickle cell disease or beta thalassemia, even if they have just SR globin. So we targeted a base editor to make this A to G change. Unfortunately, this doesn't have a canonical NGG PAM, so we had to do some evolution of Cas9 so that it recognizes non-canonical PAMs. There's more than a dozen Cas variants that are available now. If you count a bunch of different orthologs, then there's hundreds of Cas9 variants you can use with base editing. So we have now Cas9 that can find and recognize this CACC PAM that we're interested to bind. I didn't go over this in detail, but PAM stands for protospacer adjacent motif. And this is a short sequence that Cas enzymes will bind to before they'll begin testing their sgRNA for a match to the target site. So it helps Cas9 scan the genome quickly, and it helps it bind it efficiently. So the original Cas9 that was used in genome editing required an NGG PAM. And still, for most of you, if you were to order Cas9 from IDT and want to do a genome editing experiment, you usually need to, the most available version is still that NGG Cas9. So you'd be targeting a site that has NGG right next to the spacer. But now with this enzyme, we can target many more sequences. So we target this CACC PAM, we line up this guy. We're not using the original deoxyadenosine deaminase that Nicole Gaudelli had engineered either. This has been subsequently evolved into a much more active enzyme, more than 600-fold greater enzymatic activity, which leads to much more efficient editing outcomes. So when we hook these things up, now this spacer, which is equivalent to the bottom strand here, binds to the top strand, the genome, and now this bottom strand, the underlying strand here, is extruded from the double helix. It's a single-stranded R loop. So these A's are all potential substrates for deamination. Now, even though all of these are single-stranded, they're not all available for deamination because there's some occlusion of the bound enzyme. So we actually don't get deamination as high as position 16. We do get very efficient deamination around position 7. So we took hematopoietic stem and progenitor cells. These are the cells that could form an autologous transplantation treatment. So hematopoietic stem cells from patients with sickle cell disease. We treated them with our editors, either electroporating editor mRNA and guide RNA, or base editor protein and guide RNA. We saw that the mRNA was much more efficient. We got 80% correction, or 80% editing, that is, of the sickle globin allele to macrosarcoma allele. We also got very efficient editing of A9, which is in the editing window of this editor. But this makes a silent change, just converting protein to protein. Yeah, but
Is it like surprising at all, like how Cas is able to even like find the proper sequence? I mean, if there's like millions of that same pain sequence. I mean, I know you have the guide as well, but like it's 6.4 billion pieces. How does it do that sort of distance?
yeah, I'm always shocked by that as well. We know, and I think from some biophysical studies in vitro, it's seen that Cas can bind the DNA and will quickly scan through. It's a linear search rather than a three-dimensional search, which seems to increase its speed. We know also that base editors tend to be less potent than Cas9, which tends to be less potent than Cre, that has a 34-base recognition sequence. And so you need many molecules of the base editor in the cell to get efficient editing. So probably dozens of molecules per chromosome are scanning around, looking through these sequences. And we see that this can be efficient in both open and closed chromatin. There are some sites where it's less efficient, and sometimes we attribute that to very occluded closed chromatin, but Cas seems to be able, this is another surprising feature of it, it seems to be able to displace histones and other transcription factors that are bound to DNA and scan them. We know that Cas binding on its own can open up chromatin as well while it's bound. But again, it is surprising to me that this works. I never thought, when I started my PhD in 2010, I never thought that we had a tool like this that would allow us to so versatile, with such high versatility. So that's why I didn't do any genome editing during my PhD. I was only doing my postdoc in 2017 when I got into this kind of thing. So we got this very efficient editing at the desired nucleotide, silent editing next door. This is also silent at position A12. And then the first coding, the non-synonymous change that would make a leucine to proline variant, this would probably be quite pathogenic, maybe cause beta thalassemia. It was not seen because this is outside of the editing window, this editing strategy. So we got in total about 80% editing efficiency and less than 3% indels or missense variants. We wanted to know how this would look now if we were to transplant this into a patient. So we didn't want to experiment on people, as I think is good practice, so we used mice instead. First, even before the mouse engraftment, we just did in vitro differentiation of these edited cells to red blood cells. We saw that we could get switching of sickle globin, shown in gray, to macrosarcoma in blue, at a frequency that's on par with the editing efficiency that we got. And if we put these under hypoxia, we could see sickling in our unedited cells. You can see these spikes forming by polymerized beta globin, compared to our healthy cells, which stay nicely round. And so those that receive our treatments have a reduction in sickling. So this was all promising on the cell product. We looked as well at potential undesired outcomes of editing. So for base editing, this could be off-target editing in other regions around the genome, especially those that have a sequence similar to our guide RNA. So we use this computational tool, Cas Finder. Anybody can just Google this, enter in their guide, and receive within a few seconds a list of all the sites that have the most similar sequences to your guide RNA. And we also use CircleSeq. This is a much more frustrating method. It takes about a month of experimental effort to cut circularized pieces of the genome, and then all of those sites that are cut are amplified with adapters and sequence, so you can get an enrichment of places that are actually cut with the Cas9 nucleated Cas9 RNA. And so we use this to also identify where off-target edits can fall. We sequenced in total almost 700 off-target sites. We saw real off-target editing at 54 of those sites, shown by the green circle here. So 5 of those were identified by computational method, Cas-Off-Finder, but all of them were identified by circle seed. So I think this is further evidence that for initial publications of editing strategies, maybe it's enough to do a fast and dirty Cas-Off-Finder search for off-targets, but if you're actually going to put medicine into people, then I think it's important to do a much more detailed experimental off-target search. So a few of those off-target edits happened with very high efficiency. I think this is about 4 of the sites had more than 50% editing of these off-target sites. Most of them were less efficient, less than 5% or so editing at these 54 off-target sites. You can see here that the mRNA that was the more efficient editing reagent resulted in higher levels of off-target editing as well. The RNP is a more transient reagent. You're delivering the protein directly, so this protein is degraded much more quickly. And likely because of that, there's much less off-target editing. So most of the RNP edited sites have less than 1% off-target editing at these sites. We looked at where these fell in the genome. So of those 54 sites, most of them fell in the introns and intergenic space, where we don't anticipate that these single nucleotide changes would have any phenotypic impact. 4 of them did fall in gene exons, so they could lead to a coding change, but we were fortunate that these ones did not. All of them led to silent changes. And then a small number fell in the regions annotated as promoters, transcriptional termination sequences, or UTRs. But in each case, this was more than a kilobase from the closest coding sequence, and so it seems unlikely that single nucleotide changes here will drastically change the fitness of the cell. There's a lot of engineering methods that we and others are using to try to reduce this off-target editing. You can consider fine-tuning the activity. I mentioned how this this is 600-fold more active than the original one. You could make some more variants and make this a little bit less active and reduce off-target editing. You can introduce more high-fidelity variants in Cas9 to reduce off-target editing. Company Beam, that has licensed the IP of base editors, found that using inlaid base editors, where you don't tag the Cas9 with an editor that's on a floppy linker, but rather you put the animase right in the middle of the Cas9 loop, that this seems to structurally inhibit editing at off-target sites. So they tested a number of inlaid positions for the DM-based domain and saw that many of them still maintained very efficient editing at the Makasar locus, shifted the editing windows in some instances, but always resulted in less off-target editing. We also looked at our base editing strategy in comparison to a nuclease approach. So when we did this experiment, the Casgevy nuclease approach wasn't FDA approved, but this red bar shows the p53 DNA damage response that's stimulated by that now approved therapy. We track this by qPCR of p21, one of the genes that's upregulated when the DNA damage response is turned on. We get this sustained over at least 2-day activation, 4 to 5-fold activation of the DNA damage response. And this p53 response halts the cell cycle, stops cells from growing. You could envision that it might decrease the fitness for cells. In the worst case, you might enrich for cells that don't care about the p53 DNA damage response, which is one of the most common precancerous mutations you can get. So there's some concerns about this kind of outcome. In contrast, cells that are treated with our base editor approach, shown in blue, they don't activate the DNA damage response any more than a control that has no editor whatsoever. Electroporation on its own does lead to some activation, though, as you can see in the difference between the gray and black bars. Another undesired consequence of nuclease breaks is that you can get larger aberrations that are easy to really precisely quantify with sequencing. So usually when we look at the outcomes of genome editing, we PCR amplify the site that we've edited, and we use Illumina sequencing to measure that. Now, Cas9 nuclease, if you make a double-stranded DNA break, you can actually get deletions of many thousands of base pairs in either direction. And those are really hard to quantify, because if you do PCR to amplify the target site, those primers just don't bind anymore, so you don't amplify, you're sort of blind to what those outcomes are. In the worst case, you can get translocations where the target site that you've cut, the chromosome arm is distal to that is removed and stitched somewhere else in the genome. And those are even harder to quantify. So we use digital PCR here to look at the maintenance of the target site after editing with a base editor or a nuclease. We saw no change on average after a base editor treatment, but about 13% of alleles on average after nuclease treatment were missing from the digital PCR assay. So we take from this that about 13% of alleles have a larger than expected deletion or potentially chromosome aberrations. So here's the point where we're trying to test whether these edited cells can actually reconstitute into a mammal and fix the blood. Sorry, is that clock right?
It's 11:34.
That's the time that we're finishing. Well, hold on. Oh, phew. Sorry. So here we've taken our edited stem cell population. Here we just take human donated sickle hematopoietic stem progenitors. We electroporate those with our editor to treat it, and we transplant those into a mouse model that's immunocompromised, so it doesn't kill human cells as soon as they're transplanted. These cells can actually engraft and reconstitute the bone marrow of this mouse model. And we wait 4 months and harvest the bone marrow for analysis. So after a 4-month period, all of the non-stem cells that exist in our transplanted population should have differentiated and be gone. And the only cells that remain should be longer-term stem cells. We saw that at this 4-month period, we didn't change the frequency of total engraftment, frequency of leukocytes or red blood cells. So editing is not impacting the health or the lineage propensities of the engrafted cells. But we were maintaining about 68% editing. Remember, 80% editing went in, 68% editing is what we see 4 months later. So that implies to us that this mixture of CD34 HSPCs is not purely long-term stem cells. We're probably getting more efficient editing in cells that have already started to differentiate. And now, after this 4-month of engraftment, we're actually looking at editing that occurred in those long-term stem cells. So we measured 80% in the original population. We really got about 68% editing of the cells that could be the durably engrafting therapeutic. We looked at whether those cells had edits in just one allele, monoallelic edits, or biallelic edits in both alleles. We saw it was about 50-50 split between the two of those. Again, quantifying the fetal hemoglobin by HPLC, we saw a shift in the expected ratio. About 68% of the sickle globin had been converted to macrocyte globin. And cells that are taken from the bone marrow seem to show this even more drastic drop. But this is about what we'd expect for our editing efficiency, where we're now very few cells sickle. So this, we think, could form the basis of a good long-term engrafting anti-sickling treatment. One problem, though, is that human cells engrafted in mice don't actually reconstitute the peripheral blood of the mouse. So you get mouse red blood cells circulating, but not human ones. You can't really look for sickle cell phenotypes in this model where you're engrafting human cells. To look at those phenotypes, people have made mouse models of sickle cell disease where the endogenous mouse beta globin is replaced with sickle beta globin. So that's the context that we use to test the long-term phenotypic outcomes of this treatment. We took this Towns mouse model, it's called, that has a humanized beta globin locus. They have a healthy control, HBB-A, and a homozygous sickle treatment group, HBB-SS. We took stem cells from the mouse, blood stem cells. We electroporated one portion shown in blue. We left unedited control shown in red. And we also maintained a healthy treatment and engraftment. So these cells would never sickle because they have a healthy globin endogenously. This is our healthy control. We saw that over this 4-month period, we maintained editing. So about 50% down to 45% or so is the average drop we saw in edited cells. But even though we got about 45% editing on average, we saw a shift in sickle globin to macrocyte globin of about 70 to 80% by HPLC. Anybody have any thoughts about why that might be? 45% editing, but almost 80% fetal hemoglobin? That's very different than the HPLC we saw before. Can you say this?
Why do you see 40% editing with higher fetal hemoglobin? I mean, just your editing efficiency and then the expression of fetal hemoglobin are like not very independent of one another, right? So you can still have high expression of fetal hemoglobin because of, I guess, the luge or the change in that promoter regime talked about, or enhancer regime talked about earlier.
On the cell autonomous side, what we saw before was that the editing did actually perfectly match. It's now only when this is making up the blood of the mice that we see that the blood from those mice has higher. I think we don't know exactly for sure what's going on here, but we think that sickled red blood cells are very delicate. They're going to have a shorter half-life of only about 2 weeks, whereas normal healthy red blood cells stay circulating for 80 days or more. And And so cells that have been corrected have a much longer lifetime. They end up making a larger fraction of your blood than the sickle red blood cells that are dying much more rapidly. So you can get this enrichment, less editing, resulting in more healthy blood. So we did a complete blood count, which includes measurements of these parameters: hemoglobin, percent reticulocytes, red blood cell precursors, red blood cell count, and white blood cell count. We saw that in each of these scenarios, the edited cell population was much healthier than the disease one, and statistically not different from the healthy controls. So it looks like this treatment 4 months later is having a really positive impact on the blood phenotypes. We looked at sickling in these cells from mouse blood and again saw a big drop in sickling propensity. We looked at the editing outcomes. And again, these mice now have been engrafted for 4 months, and about half of their red blood cells have edits on both alleles, half edits on one allele. I think it's important to note this allelic heterogeneity, but we don't really have a clear sense of how this can actually change the outcome. Either biallelic or monoallelic cells should support a healthy hematopoietic system. I think the key thing is maybe the 20% of cells that have no edit at all. This is the portion of cells that should still sickle just as badly as there's no treatment. We also looked at the morphology inside the spleen. So when you have this rapid turnover of delicate red blood cells, the amount of red blood cells your bone marrow makes is no longer sufficient. So your spleen starts to differentiate red blood cells as well. So in those mice receiving unedited sickle stem cells, they're getting this extramedullary erythropoiesis, the differentiation of red blood cells in the spleen. This is marked by an expanded red pulp in the spleen. Those mice receiving our edited cells do not have the splenic pathology, so they look much more similar to healthy control. You also get this pooling of blood in the spleen that leads to splenomegaly, or this vastly enlarged spleen in your disease samples. And those mice receiving our edited red blood cells had a substantial reduction, more than halfway towards the size of a healthy control. So on average, we're correcting a lot of different phenotypes by grafting our edited stem cells. We did a little bit more. We tried to dig in and look at exactly what editing efficiency we need to correct the hematopoietic phenotypes here. So we took cells that had already been grafted into our into these mice. So these mice showed healthier blood. These are negative control mice that showed unhealthy blood because they didn't receive any edited cells. We mixed the edited and unedited blood from these cells in different ratios, ranging from 100% of one to 100% of the other. And we engrafted each of these ratios into 3 new mice. So the idea is that this is a secondary transplantation, taking cells from the primary recipients and injecting them into a secondary recipient to see how much editing you really need to correct the phenotypes. We saw that editing in these secondary recipients was lower on average, but still visible, and it tracked with the percent of edited cells that these were given. And here I'm showing those parameters that we saw from the complete blood count, the percentage of the total concentration of hemoglobin, percentage of reticulocytes, red blood cells, and white blood cells. In each case, we needed about 20% editing, the light blue bar here, to get restoration of the phenotype close to a healthy control, marked by the dotted line. So it's a little bit surprising that only 20% editing can correct the majority of the blood phenotype. This is actually a concept that we see in several different contexts of genome editing therapies, that you don't need to correct 100% of the tissue to restore health. Sometimes you may just need to give the cells a nudge back into a healthy state. The body already does so much to maintain your health that complete editing efficiency is really needed. One of the papers I think we'll be discussing later this week involves base editing that will induce fetal hemoglobin rather than modifying the sickle cell mutation itself. I won't go into detail about that now here, but I just wanted to give this one slide overview that these involve base editing guides that we target to the fetal hemoglobin promoter, so the sequence just upstream of fetal hemoglobin or gamma globin. If we make these nucleotide changes, we're introducing new transcription factor binding motifs. All of these variants were known to us before doing this study, so these are naturally occurring in the human population, and patients that inherit sickle cell disease as well as one of these variants are protected phenotypically because now they're expressing more fetal hemoglobin. We saw that when you look at the potency of each of these edits compared to Cas9 approaches to disrupt BCL11A, so this is the FDA-approved therapy, base editing to introduce TF binding sites can actually be much more potent on a per edit basis. So I think there's more interesting discussion that I will have later this week on this kind of thing. So I talked now a lot about base editing in the context of sickle cell disease. I'm going to touch on the other editing method that has been shown to now be efficient in vivo. It has a much wider scope of potential editing outcomes. This is prime editing. So prime editing doesn't use a deaminase to chemically react with the DNA. Instead, it uses a reverse transcriptase to write new sequences into the DNA. So you still use Cas9 to find the target site. You still use a nickase to nick one strand. With base editors, you're nicking the strand that's bound to the guide. With the prime editor, you're nicking that R-loop strand that's not bound. And the key change with prime editors is not only the reverse transcriptase that you fuse, but also the sequence that you encode in the guide. So for for base editing and nuclease editing, you're just changing this 20 nucleotide sequence down here. For prime editing, you're also encoding this 3' region that contains binding site for this R loop, and it contains all the information you want to write into the genome. So you nick, you anneal this 3' primer binding site, and then the reverse transcriptase will directly write into that nicked genomic strand any sequence that you want that you've encoded onto the genome that you've delivered. So you're writing a totally new sequence right into the genome. When the prime editor falls away, you're left with this equilibration. Either the original endogenous genome can be annealed and all the newly synthesized sequence is extruded from the double helix, or you can have annealing of your newly synthesized sequence into the genome. You always have to include some DNA sequence that's precisely matching your genome after the edit that you want to make, specifically so you get this outcome where that newly synthesized sequence is annealed back in. And so this is more thermodynamically stable, clearly, but cells like to cut off 5' flaps because that's something they do all the time during during DNA replication. So very frequently you'll get processing of this 5' flap, resulting in this mismatched double helix. So at this point, you can get DNA repair in either direction. You can eliminate your new edit and restore the endogenous sequence, or you can cut out the endogenous sequence and prepare it to contain your endogenous edit. Either of these outcomes are seen, but we know with prime editing there are different ways you can bias repair to the way that you want that encodes the edit. And one of the main ways to do this is to use that same trick we found from base editing to nick the unedited strand somewhere downstream of this site. To do that, we usually add an additional guide RNA to the mixture. But the main point is you can nick the optical strand, greatly enhance the rate of the desired editing outcomes. So in addition to making transition changes like the base editors, now you can also make transversion nucleotide changes. You can also make precise insertions of tags and deletions of long unwanted DNA sequence as well. And unlike Cas9 nuclease, where you get uncontrollable mixtures of these insertions and deletions, now you can have a very pure editing outcome with precisely the number of nucleotides changed that you want. So now for the first time, you could, for example, treat that Tay-Sachs disease caused by insertion of 4 nucleotides just by precisely deleting those 4 nucleotides to repair the gene. Any questions about prime editing mechanisms? There's a lot to talk about here. This was first developed in 2019, also in the Blue Lab while I was a postdoc there. And I'd say there's been about a dozen papers every year since then trying to improve prime editing and engineer it in specific ways and do tricks to get this to be more efficient.
So is there like, does it really matter where that nick occurs on the R loop? Like, does it have to be within like 3 bases of the end of the loop, or is that just, schematic?
So Cas9 nuclease, pyogenase Cas9 nuclease, the variant that we use typically, will generally make a double-stranded break 3 nucleotides away from the PAM. And we inactivate one of those so that we're not getting a double-stranded break, because that would result in those uncontrollable insertions and deletions. But we keep one there, so that does cut 3 nucleotides upstream of the PAM, generally. There's a little bit of fuzziness where it can chew back a little bit more or less, but really you can only count on it nicking right here. And that's really important for the prime editing strategy, because if you're writing in sequence after that nick, we're not able to edit anything upstream. So remember, this region right here is the part that gets base edited, but we can't actually change those nucleotides with prime editing. We can only change nucleotides downstream of the nick. So the positioning of the nick is really important. Though you could base edit these nucleotides, if you wanted to prime edit them, you'd need to use another guide RNA that points in the other direction, right sequence, to rewrite the target.
Do you just have to rely on the repair mechanisms to be kind of random when you're doing the insertions of the genes, or even after the flap to correct the other side?
So if you add this additional nicking guide, then you can nick the unedited sequence, and then that helps. But it is still random. Yeah, there's a lot of complicated factors involved in repairing DNA mismatches. I think it's not possible to try to link them all to the editor and enrich them at the target site. So we haven't seen anybody add more than a couple of domains. But we have engineered some mismatch repair inhibitors that increase the frequency of reparative side outcome. Those, in some sites they really help, at other sites they don't.
And how long can be added right now or adjusted?
People have shown efficient editing of 200 base pair insertions. That's still really hard to achieve, so I think that people do a lot of screening to find efficient editing strategies. That's sort of the maximum I'm aware of. The very first paper showed 40 nucleotide insertions, 80 nucleotide deletions. I think that's still the limit of what you can expect to do efficiently. But in my lab right now, we're trying out some other approaches that could get us longer sequences based on other publications of people who have achieved that. Good questions.
How do like the variants, I guess, for prime differ compared to like for the base editors? They have like different PAM sites that they can recognize.
Yeah.
Is that possible for the prime editing scenario?
Yeah, some people have shown that. So one issue is that prime editing tends to be a little bit weaker at binding the target site. So the PEG RNA itself that has this 3' extension doesn't bind as well to Cas9. And so it really knocks out editing efficiency if you have lower binding of the guide and a Cas9 variant that's weaker binding to the DNA. So the most common thing, say more than 90% of all prime editing uses wild-type biogenese Cas9 binding to an NGG PAM, because that binding is very tight and specific. And the reason why that's possible with prime editing but not with base editing is that with prime editing you have a lot of versatility to rewrite almost any sequence you want, 40 base pairs down the target site. With base editing you have to really align this with the target nucleotide and no bystander nucleotides are in the editing window. So some people have shown, there are cases where there's just no NGG PAM around the target, so you can use Cas9 variant that binds to a non-canonical PAM. It's just on average less efficient. And so this 40 to 200 base pair insertions can correct more than 90% of pathogenic variants, but it's not good enough to write in a whole new gene sequence because genes are often many kilobases long. What you can do is insert a recombinase binding site that's 30 or 40 nucleotides and then recombine in a whole healthy gene. So there's a lot of research going into that kind of approach as well. But I want to show that we had also used prime editing to correct sickle cell disease. So this now, instead of making macrosarcglobin, the rare but healthy variant, we can restore HbA, the common healthy beta globin variant. So we received cells from 4 different donors here, electroporated them with our prime editor, mRNA, and PEG RNA. We got, the worst case, about 16% editing, and the best, about 40% editing after the engraftment into mice. And this did result in loss of sickling, about 20% editing, which we think is around the threshold. I think this is still a little bit too close for comfort. We published this in 2023, and since then there have still been a lot more improvements on prime editing methods. So I'm really hopeful that there'll be even better editing efficiencies we can achieve in the coming years. Actually be able to correct beta globin back to wild type. This is just showing the drop in sickling. So in proportion to the amount of editing that you're getting, you're also decreasing sickling in cells. With prime editing, there's many steps that require the hybridization of DNA to your guide RNA. And so it's particularly specific. So I remember I showed with the Macrosar editing strategy, there were 54 sites that actually had off-target editing. We screened many sites with prime editing using CircleC and didn't see any statistically significant off-target editing. There are some sites, so gray is unedited cells, yellow is edited cells. So there are some sites that are noisy and look like they're edited even without any prime editor going in there. But on average, none of these are statistically significantly edited greater than the unedited control. And you can see the axis here, none of this even rises above 1% modification. Now we used a nicking guide here that will target the opposite strand for nicking to enhance our editing efficiencies. And there was one region where we saw statistically significant increases in indels based on, we believe, off-target nicking. And this is actually because the off-target site here is the delta globin gene, which which is located in the beta globin gene cluster. So now we're nicking multiple places close by one another, and that leads to this about 1% deletion of the intergenic space. Here's the reason why we think prime editing is so specific. So nuclease and base editing is specific because of this first binding event between the spacer and the target genome. Prime editing also requires this PBS interaction where your guide RNA hybridizes with the R-loop strand. So that's the second place where a mismatch could prevent editing outcomes. And then third, we need this hybridization of the newly synthesized strand to the endogenous genome downstream of the make. So we're not only looking at the guide RNA, but also all of the nucleotides are synthesizing downstream of that. So if you don't have homology here, then you won't get editing outcomes. And so all three of these together seem to vastly decrease the frequency of prime editing on targets. So if we want to use this for in vivo delivery and treating many more diseases than blood disease, we'd like to formulate these in a way that doesn't require bone marrow transplant, which is directly inject therapy. So not only for other diseases, but even for correcting sickle cell disease, we don't want to have patients go through this painful bone marrow transplantation every time. We'd rather just be able to inject the editor into the blood and correct it. There were just 2 publications in the last year or so from UPenn and MIT that reported nanoparticle delivery of editors to the blood. However, nuclease editing efficiency and base editing efficiency remains really low. Shizhen Lian, who's now at Hopkins as well, published another one just recently. In each case, it's only about 5% or less editing in the blood, but I still think this is a strong, promising start, and we're soon going to see better editing strategies for these targets. One last editing strategy I want to show for the blood is the use of adenoviruses in selection. So I mentioned before, adenoviruses cause this big immune response that ended up killing Jesse Gelsinger. People have now evolved low immunogenicity variants of adenoviruses, and now that we know to add immunosuppressants, then we expect we'll be able to stop that kind of death in the future. And so it revives this possibility of using adenovirus to correct genetic material. This is a group, we collaborated in the Lou Lab with Andre Lieber at Seattle. They showed that they can inject an adenovirus in vivo into that Townes model of sickle cell disease. And they'll get a very small amount of editing in the hematopoietic stem cells. But along with the editor, they're delivering this selectable marker, which is a variant of the MGMT gene. It causes resistance against chemotherapy. And then they add this chemotherapy drug, 06BG, for 3 different doses. And they see that they can enrich for less than 1% editing at the get-go. Now they're killing off the endogenous stem cells slowly over time, and by 4 months, they're getting over 40% editing in the blood. So there are interesting tricks like this. I should say this dose of chemotherapy is actually 5 or 10-fold lower than what's used in an actual patient for chemotherapy. So this is a much lower toxicity threshold of dosing. This should be much more comfortable for patients, and still results now in the long-term hematopoietic system being made of biallelic or monoallelic edited cells. Sickle cell disease is the first and most visible target because of its wide prevalence. But if these treatments look promising, we expect we can apply the same editing strategies to many other blood disorders. While blood is particularly amenable to extraction, modification, and retransplantation, there's a lot of diseases where you can't take out the organ very easily, like the brain or the heart or the liver or the lung. And so we'd like to develop in vivo delivery strategies like those using adeno-associated viral vectors or lipid nanoparticles to edit and treat more disorders. And I talked about many clinical trials where that's now being tested. I wanted to show a little bit more data how that looks. So we can insert our base editor mRNA into a lipid nanoparticle, deliver that to the livers of mice. We get about 60% editing by day 7, which is maintained stably even 100 days after injection. Here we're targeting ANGPTL3, which is an inhibitor of the lipases that degrade cholesterol. So destroying ANGPTL3 should lower triglycerides and cholesterol. We do get more than 90% drop in ANGPTL3, shown by this triangle dots here, and reduction of 50% in the cholesterol as well throughout the 100 days in these mice. So we think this would be a promising approach. Something very similar to this is in clinical trials right now. In addition, we saw we could actually get very efficient multiplex editing of both ANGPTL3 and PCSK9, both of which can prevent cholesterol buildup. So there's opportunities to correct many genes in the liver of mice at the same time. What I want to mention here is about 60% of the liver is hepatocytes. The rest is stellate cells, macrophages. And so when we're getting about 60% editing, we think this is almost 100% of hepatocytes that are changed to have the desired genotype. Another tool, I'll go quite quickly now since I know we don't have very much time left, but I want to highlight that there's other delivery modalities that are being explored now. Using engineered virus-like particles. These have been changed so they no longer encode any nucleic acid, rather they encapsulate the editor proteins themselves. So original forms of these were inefficient, but by conducting a lot of engineering on the on a protease cleavage site between the capsid and the editor link, then we can get this more and more efficient. And now, using this tool, you can deliver protein. The DEVLP is a protein delivery rather than plasmid. This can allow you to get even more efficient editing of the target site and prevent off-target editing that you see from sustained expression of the editor over plasmids. Now, this protein is only around for a day or so, and it doesn't even have enough time being in the cell to edit these off-target sites. We showed that this can very efficiently transduce and edit the liver, and also be injected subretinally to edit the eye. This restores blindness in that Leber's congenital amaurosis mouse model. We also used a base editor in vivo in a mouse model of progeria that's characterized by this rapid aging. Patients usually die in their early teens. Encoding these base editors in viral vectors, you can get about 30% editing in the heart, quadricep muscle, aorta, and bone. And again, about 60% editing in the liver. And this is enough to fully prevent the pathology in the aorta. So that there's a loss of smooth muscle cells in these mice that are affected by progeria. Those are totally restored in edited mice. There's also the expansion of this thick adventitia, this fibrosis that prevents the aorta from working normally, and that's totally prevented in mice that receive this. I should say all of this is 20% bulk editing in the aorta, but almost complete correction of the phenotype. So again, just nudging the body into having that little bit of a healthy genotype is enough to let the body correct itself. Here's a video I want to be sure to show you of a mouse that's nearing the end of its life. We have to euthanize this mouse. It can't really move. It has matted gray fur and sort of shivering and shaking. It's very ill. But mice that received one dose of AAV 2 weeks of age, now this is 4 months later, and these mice are still running around much more happy. So we can hopefully do this in patients, extend both their lifespan and their health span. So in total, these mice that otherwise die around day 200 now live to around day 500, 2.4-fold extension to the amount of time they had healthy. I'll skip through this. This is just showing similar treatment that prevents cardiomyopathy and can be used in the brain as well to edit SMN2 and treat a spinal muscular atrophy model by gene editing. I also wanted to reiterate that there's many other gene therapies that target cancer or autoimmunity, but here I'm not focusing on those, I'm just focusing on genetic disease. So what we really need in this field are safer and more efficient in vivo delivery technologies, editors with even more improved specificity and efficiency, and we want to also be able to assess long-term administration and impacts of these as we're comparing between many different candidate therapeutics for sickle cell disease and other genetic disorders. I think it's a really exciting time for genome editing for us. I hope that many of you will also contribute to getting better and more effective treatments for genetic diseases in ways like this. This is just summarizing what I said already, but I just want to highlight many people that led or co-led the studies I showed, and also thank David and Luke for contributing some of these slides. Thanks, everyone. We're out of time, sorry for going a little over there. If there's anything you'd like to discuss, feel free to move to your next appointment.
Does proximity to like a TAG affect editing efficiency?
Proximity to a tag? To a tag.
Yeah.
Proximity to a topologically associated domain?
Yeah.
I mean, everything is part of a topologically associated domain, right?
Or just like CTCF binding sites in that form.
I thought the whole genome is oriented into those loops, so anything that you do will be, you mean proximity to the CTCF binding site itself? That's a good question. I haven't seen any studies that really explore that in good detail. I wouldn't expect so, because my understanding is that those loops are an average of many cells, but they're dynamic in how they're formed. So I would suspect that Cas9 would be able to bind whenever those loops are transiently disrupted and do its thing during those times. So I wouldn't expect there to be a significant change based on how close you are to the edge of the tech upper. But yeah, it's a bit, it would be interesting. So I guess I'll see you on Friday then for some paper discussions.
Thank you, Greg.